Well, in order to sort of discuss that, we have to talk about the conduction of electricity. So let's do that real briefly. Because this tissue, like muscle, can conduct electricity, and again, low voltage, but electricity nonetheless, we'll just take a look at a cell. Cell membrane, all right? Remember from chapter 3, what the cell membrane is made up of. You have an outside of the cell, and you have an inside. Okay, and these cells, save for the cells on the extreme surface of our skin, all of the cells in our body, which make up tissues, which make up organs, and so forth, they're bathed in water. All right, now we have these ions that are responsible for the conduction of electricity. The primary ion on the outside of the cell is the sodium ion. Okay, and I don't know if you remember from chapter 2, but the chemical symbol for sodium is Na. It's not S because S is used up by sulfur, so we use the Latin name for sodium, which is natrium. All right, so the primary ion outside is sodium. And when they draw your blood and they're checking your Chem 6, all right, they look for your sodium levels. Average 135 to 145 milliequivalents per deciliter. Now, there are some potassium ions, but very few in comparison. The primary ions inside the cell are the potassium ions. Now, there are some sodium, but fewer. But you also have a lot of negatively charged ions, like phosphate and sulfate. I think it's 3. Okay, so when we look at a nerve cell, like a muscle cell, we look at it in a steady, unexcited state. And we have this distribution of ions, primarily sodium on the outside, potassium on the inside. And there are proteins in the cell membrane that use energy to maintain this distribution, this homeostatic distribution of ions across the cell membrane. Keep most of the sodium out, keep most of the potassium and these phosphate ions in. Okay? Now, in this state, unexcited, no stimulus yet, we call this state a resting state or a resting potential. And we say that in this condition, the cell is polarized. Okay? Now, I don't know if we discussed this before when we talked about muscles, but polarized, what does it mean? What does polar mean?
Two ends.
Two ends, separated by something, right? Yeah. The poles on the Earth, north and south, are separated by a big ball. Okay, if you go out and spend a couple $100 on a really good pair of polarized sunglasses, which if you're a fisherman, you would do that, because if you're out on the ocean, the light is bouncing off the water and causing all sorts of reflection and glare. What polarized sunglasses do is they separate out certain wavelengths of light so that it reduces the glare. Polarized is that we have a separation of electric charge. All right, in this resting state or the resting potential, we say that the outside of the cell is positively charged, and then for ease of understanding, we say that the inside of the cell is negatively charged, when in truth, the inside of the cell, because of the presence of a lot of these negatively charged polyatomic ions, it has a less positive character than does the outside with all of these positively charged ions. So we just, instead of saying, well, the inside is less positive than the outside, we'll just call it negative for ease of understanding. And this voltage drop in neurons across the cell membrane is somewhere in the neighborhood of a minus 70 millivolts. Okay? Now, what happens when a nerve cell gets excited or gets stimulated? And it can get stimulated by some physical means. In other words, if I touch you, I stimulate the neurons. Or there can be a transmission, that electrochemical event. One axon or one neuron releases a neurotransmitter onto another, and it causes a reversal of this polarity. All right, so let's just hold that thought for a minute. First, in the resting state, the resting potential, the cell is polarized. The outside has a much more positive character than the inside, so we'll say it's positively charged. The inside has a less positive character, so we'll say it's negative, just to try to get ease of understanding. Now, what stimulates a neuron to conduct electricity? All right. In this case, we'll say some neurotransmitter gets released. Okay, and there are many different neurotransmitters. And let's say there's a protein receptor here in the cell membrane, just like in the skeletal muscle. Here you've got this neurotransmitter coming along. It binds to the receptor, and that binding causes that receptor to change its shape a little bit. And what that does is that allows then for this influx of sodium ions. So for a brief time, and I'm talking about milliseconds, the sodium ions move into the cell because of diffusion. You know, diffusion is movement from an area of high concentration to an area of low. And since there's much more of that stuff out here than there is in here, it's just going to move in. And an effusion or efflux, if you will, of this potassium ions. All right, and then for a brief instant, the cell becomes depolarized, which is actually a reversal of polarity. So for that brief moment, the inside of the cell becomes more positive than the outside of the cell. And this sets up a series of events. The depolarization in, say, this section of the nerve cell membrane causes certain of these cell membrane proteins, these voltage-gated ion channels, to open. Sodium goes in, potassium goes out, and this continues all the way down the length of the neuron cell membrane till it reaches that, that end bulb. Okay, now, that takes milliseconds. We break it down into, this gets depolarized, we get this change, this reversal of polarity, which causes this part to become depolarized. With this reversal of polarity, this gets repolarized immediately because of these, what they call the sodium-potassium pumps, which reestablish the distribution of these ions across the cell membrane. And then this part gets depolarized, this gets repolarized, this part gets depolarized, this gets repolarized, and it sets up this wave. Okay? And again, it's milliseconds.
Just like that exercise we did last week for the nerves.
Yeah. Okay. Now, what will cause this reversal of polarity is called a threshold stimulus. Because, sometimes you'll receive incoming stimuli, and it won't result in a so-called, this whole thing is known as an action potential. Is reversal of polarity and then a reestablishment, reversal, that's called an action potential. And once it starts, it doesn't stop until it reaches the end. It's the so-called, it exhibits what is referred to as an all or none response. Now, you might ask, well, what determines whether it is going to be an all or none response, or if there's going to be any sort of action potential generated? And what different nerves, but I believe in the nerves in the brain and in the spinal cord, this reversal of polarity has to take this voltage potential, this voltage drop, and increase it from -70 up to, I believe it's about a positive 55 millivolts. And that is dependent upon how much of this sodium and how many of these voltage gated channels actually open. All right, if, just for argument's sake, we'll say that it's going to take 10 of these voltage gated channels to open to allow enough sodium in to bring this from -70 to positive 55. If only 8 of them open, because of the stimulus, the intensity of the stimulus, or then no, we're not going to get an action potential. Isn't going to happen. Not enough change occurred. We did not reach the minus 55 millivolts. We did not reach the threshold stimulus. All right? So, who cares about all of that? Well, what I want you to remember about this is, 1, the resting state or the resting potential. In an unexcited nerve, we have a resting potential, and it involves a voltage drop across the cell membrane equivalent to about -70 millivolts. Okay, I don't know, and we don't really need to go into a discussion about what voltage is and so forth, but it involves electricity. Okay? Now, if we have some stimulus that occurs and enough of these sodium channels open and enough of this sodium, because it's positively charged, so we're going to take this negative value and raise it to a positive value because of these positively charged ions. If enough of them go in, and we raise this up to some threshold, which I believe in this case is about positive 55, then bango, we have this action potential that will travel down the length of that axon. Okay, it's called the all or none response. Okay, it's either gonna happen or it isn't. If you don't reach plus 55, it ain't gonna happen. If you do, then it will, and you'll get some response. Okay? The important terms: polarized, depolarized, and then repolarized. And that repolarization, in other words, we're reestablishing our resting potential, is carried out by these so-called sodium-potassium pumps, which actively transport things against a concentration gradient. Okay, all of this sodium came in, so now there's more here than there is out here, but we want to move this out here, so we've got to move it uphill. Takes energy. Okay? And now, we don't really... Potassium, on the other hand, is always kind of leaking through the cell membrane. And so this sodium potassium pump is sort of always acting to keep the potassium levels at homeostatic parameters across the cell membrane. Okay. And I'm sure you're aware, low potassium, not good, right? You can experience things like muscle cramping. Your muscle cells aren't going to conduct electricity right very well, okay?
All these energy drinks have potassium in them?
I'm sorry?
These energy drinks have potassium?
Oh, yeah. What do we use in the Commonwealth of Virginia to execute?
Sodium pentothal.
Well, they do that out of mercy, but what do they actually use to kill them? Potassium chloride. Very high doses of potassium to goof up this whole business, this whole resting potential, and it actually stops the heart. Okay? Not a pleasant thought, but anyway, take home message. We have a polarized state, then we'll have some stimulus, and the stimulus is either going to open up enough of these sodium channels to bring us to threshold. If so, we're going to get an action potential. If not, we won't. If we get an action potential, we have these proteins, these sodium potassium pumps, to reestablish the concentrations of these ions across the cell membrane, and the cell becomes repolarized. Now, for a brief time, there is a phenomenon known as a refractory period where you can stimulate the cell again, but because it hasn't repolarized, you're not going to get another action potential. So it's unavailable. All right? All right, good. All right, now, because I don't want to get hung up on a lot of these, we have, all right, I've sort of already alluded to this. If we look on page 272, you're going to see a description of the so-called synapse down there, okay? And this will help to sort of reiterate or drive home the electrochemical nature of the transmission of nervous impulses in the brain, spine, the central and peripheral nervous system. You see at the end of the neuron you have an end bulb, okay? And then you have a synaptic cleft, which is a space, and then you can see where a neurotransmitter gets released over here from the presynaptic neuron, the one that comes before the synapse. Those neurotransmitters bind to some receptor in the postsynaptic cell membrane, and then we get this business happening, this depolarization. Okay? And you can see there'll be a change. They make, now, I guess I will bring that up. An important ion that's involved in the release of this neurotransmitter is calcium. All right? When the action potential reaches the neuron end bulb, causes a change in the permeability of the membrane of this neuron end bulb to calcium. Calcium ions will flow in. That will result in some of these little vesicles, these sacs that contain the neurotransmitter. They'll fuse with the membrane, and then they'll dump their contents into the synaptic cleft. All right, now, once the neurotransmitter has been released, one of three things can happen to it. All right? One of them is that it can be enzymatically decomposed. Like if you remember from the muscles, there was an enzyme on the muscle cell membrane called acetylcholinesterase, which broke down the acetylcholine released by motor neurons. More so, I think, in the brain and spinal cord, what happens to this neurotransmitter is, one thing is that it just diffuses away. because you have a high concentration here, none over here, and it'll just move because things diffuse because they have inherent energy. Okay. A lot what happens is there's an active transport process where this neurotransmitter is taken back up into the end bulb in a process called reuptake. And again, it's an active transport mechanism, and then it can be recycled and reused. It's not Paxil, what's the drug out there for depression that everybody's using nowadays? Used to be Paxil.
Adderall?
No, that's a stimulant. For depression, it's for mood.
I know what you're talking about, and I can't even think
It's not
of
coming
it.
to me either.
Anyway, we'll use Paxil because it's the same class of drug. But there was back in the 80s, I believe, or 90s, Paxil was the drug. Okay, and what Paxil is, its class of drug is called an SRI, a serotonin reuptake inhibitor. And what that drug does is it sort of interferes with this reuptake of the neurotransmitter serotonin. Now, serotonin is a neurotransmitter that's involved with mood. Okay, so if we block that reuptake with this drug, this neurotransmitter will stay in the synapse longer, and we will feel better, or we won't feel depressed. What is the name of that?
I'm working on it.
It's just it's a common drug.
Come to me. Yeah,
Right,
just
everybody
search that
takes
through.
it. I just can't think of it. I'm having a brain spasm here. Anyway, what it blocks is this process.
Zoloft, Celexa, Prozac.
Prozac. Everybody takes Prozac. If you're feeling bad, take Prozac. Prozac is what is called a SRI, a serotonin, and it's specific for that neurotransmitter, reuptake inhibitor. So the longer that serotonin stays in the synapse in our brain, at least theoretically, the better we feel. Okay? All right. So, resting potential, action potential, polarized, depolarized, repolarized, and then the fate of these neurotransmitters in our brain is that they can diffuse away, they can be decomposed enzymatically, or they can be retaken back up into the synaptic, the presynaptic end bulb, and then to be used again. Okay? All right. Because we got to move and we got to keep on moving. And if you look at figure 911 on page 274, you'll see that there is a protein in the cell membrane. Okay? And that says channels in the cell membrane partially determine the distribution of ions. These ions that we're talking about, sodium and potassium, inside and outside the cell. All right? So you sort of have a brief look at what an action potential is, what a resting potential is, and what a threshold potential is. If the stimulus that happens, whatever that stimulus is, if it doesn't cause the membrane to reach threshold, there will be no action potential. If the action potential does occur, it will occur in a all or none response. All right, let's move on. Now, that's kind of explained in a little bit more on page 278 and 279. You'll see the membranes there, and the sodium on the outside, the potassium on the inside, and then you can see this wave as it passes down the cell membrane in the so-called all or none response. All right? All right, now, and again we have hundreds of different types of neurotransmitters, and depending upon where they are, they have different effects on the body. They can either excite a postsynaptic membrane, postsynaptic neuron, or they can inhibit a postsynaptic neuron to block any more electricity from going down there. Okay? And we don't need to try to differentiate all the ones that are excitatory with all the ones that are inhibitory. Just know that there are those two different types. Okay? And depending upon the different neurotransmitters are excitatory. Some neurotransmitters are inhibitory. And there's a table, if you look on page 280, it says, for instance, a type of neurotransmitter known as a monoamine, such as norepinephrine, okay, creates a sense of feeling good. All right, dopamine creates a sense of feeling good. Serotonin, okay, primarily inhibitory. All right, leads to sleepiness. And then it says the action of serotonin is blocked by LSD. I don't know if you even know what that is, but it's very popular. Not that I ever did. It was very popular back in my day. Okay, you can see GABA, these amino acids. And I'm not going to ask you to memorize or, the different types of neurotransmitters. The point is that some can be excitatory and some can be inhibitory. Okay? Now, this idea of convergence and divergence, as the names they're sort of self-explanatory. If you have a convergent situation in the brain, you have several different neurons converging on one. Okay? If you have a divergent situation, you'll have one neuron branching out and stimulating several other neurons. They diverge out. Okay? We don't need to know a whole lot more about that, but we do need to know these terms, and we need to make sure we know these terms. You have afferent and you have efferent. And these are explained, and we'll just look at them briefly. Types of nerves, okay? It says bring sensory information into the central nervous system. Sensory fibers are afferent fibers. So they bring information into the CNS, or the central nervous system. Okay, these are afferent fibers. The efferent fibers send information out from the CNS. To the rest of the body. Afferent and efferent. And you'll see these terms recur in other body systems. Like, for instance, in your kidneys, you have afferent arterioles that bring blood into the kidneys and the nephron, where blood gets filtered, and then you have efferent arterioles, which take that blood after it's been filtered out of the nephron. So afferent brings something in, efferent sends something out. And I would become familiar with those because, again, they will recur in other body systems. Okay? Now, in our spinal cord, it's arranged in such a way, and I want you to think about this. When you think about nerves, the term nerve, don't think about a single nerve cell, because a nerve, particularly the ones you can see, if you were to cut yourself deep enough and you could see some white matter down in there, a nerve is actually a bundle of, hundreds of thousands of different individual nerve cells. All right? In the spinal cord, we have nerves that go from our brain, down the brainstem, out the spinal cord, to the rest of the body, and then we have nerves that come from the sensory, those are efferent neurons. Then we have sensory neurons that take information from out in the environment, stimulate us, and send that information up to our brain. And you'll see, the spinal cord is arranged in these bundles of nerves that are called tracts, nerve tracts. And they all have names, and they're named according to where they originate and where they end. And we're not going to get into a lot of those because I want you just to have a basic picture of this. But the way that nerve tracts are arranged is that, and there's a lot of exception, but typically the outside of the nerve tract will be sensory nerves, and then the inside of the nerve tract will be motor nerves. Now, how do I know this? Well, let me tell you. I've ruptured multiple discs in my neck. Okay, and then when the disc ruptures, and you'll see that when we look at this vertebrae, but it will, the nucleus pulposus will rupture through the annulus, and it'll put pressure on a spinal nerve. All right, so a little bit of pressure, probably all you're going to feel is pain or some unusual, not normal type feeling, because the outside of these nerve tracts are sensory. Okay, but then after a while, like the first time it happened to me, I felt a pain right under my shoulder blade. And I had been out cutting wood and doing things I do out in the yard, so I didn't think anything of it. And a month or so went on, and I guess the rupture got worse, and the pressure on that nerve got worse, and it started pushing in to the inside of that nerve tract, and I started to lose motor function. And it got so bad that I couldn't pick up my infant child with my left arm. All right? So just what's all that about? Well, just know that these nerves are actually bundles of, hundreds of thousands of different neurons. And as a generality, they're arranged sensory on the outside. And I'm talking about going from the brain to down the spinal cord, and from the periphery out coming back into the central nervous system. Now, there's always going to be exceptions to everything. So that's just a generality. All right. You can see there, we on the same page, on page 287. Just take a real quick look. You'll see two types of tracks. Ascending, you see that there? Ascending tracks, which are going to bring information up to the brain. And then descending tracks, which will take information from the brain out to the periphery. Ascending tracks will be afferent. Descending tracks will be efferent. All right. We're not going to have a lot of time to go into a lot of the different sections of the brain, but just know that